Hello and welcome to this podcast for cell biology. In this podcast, we're going to continue our discussion of the cell cycle. But in this podcast, we're going to focus on how the cell cycle is regulated, so regulation. And then either towards the end of this podcast or maybe a separate podcast, we'll talk about cancer and how a dysregulated cell cycle can lead to cancer. As we get started, I want to remind you we have our cell cycle here with the M phase. Once those two new cells are produced, it will enter G1 phase, and then S phase where the DNA is copied, and then close with G2 phase. Within an organism, we have a wide diversity of cells that divide at different rates and signals. Some signals may cause some cells to divide, but not others. Because remember, we wouldn't want this cell cycle to be on all the time, and we don't want it to be off all the time in all cells. For instance, as we've talked about before, neurons and muscles don't divide. They exist in this part of G1, which we didn't mention before, but we'll mention it here, called G0. They exit the cell cycle, and they never return to the cell cycle to divide anymore. So they don't divide. What tells these cells not to divide? Then we also have cells like embryonic stem cells, which divide on average about every 30 minutes. So two wide ranges here, don't divide and divide pretty rapidly every 30 minutes so we have these two extremes within an animal. You have neurons and muscles that don't divide, and then these embryonic stem cells that divide really rapidly. And then we have cells that are somewhere in between, like a fibroblast. That divides every about 20 to 24 hours. And then we have skin cells, which divide fast, but not quite as fast as embryonic stem cells. So they're about every 11 hours. So there's got to be some way to signal a cell to divide or not to divide. So scientists knew that there had to be some regulatory mechanism. And we'll talk about the different mechanisms found throughout the cell cycle, but I want to begin with this transition from G2 to M, because that was the first factor discovered that suggested there was actually a mechanism, a molecular mechanism that regulated the progression from one phase of the cell cycle to the next. So let's talk a little bit about those experiments. The first set of experiments involved studies that use cell fusions. So what they did in these cell fusion experiments is they took a cell that was in M phase, that had a nucleus that was in M phase. They then took this cell and they fused it with a cell in G2 phase, also of course containing a nucleus that was in G2 phase, meaning that the DNA had been copied and was ready to enter M phase but had not quite done that yet. And when they fused these together, what they observed was the following. The nucleus in the M phase cell remained unchanged, but the nucleus that was in the G2 phase cell quickly entered M phase. Now, one could have argued that the reason it did this is because it was ready to. It was at G2, remember, and G2 is ready to go into M phase. So maybe that's why it did it. However, what happened is when we took this M phase cell and fused it with either a G1 cell with a nucleus that was in G1, so the DNA had not been replicated, or a cell that was in S phase containing a nucleus that was in S phase, so the DNA was in the process of being copied. So when they made these fusions, what they discovered was the following. The M phase cell fused with the G1 phase cell had no effect on the M phase nucleus, remained in M phase, just like up here. I should have wrote that out. But the G1 phase nucleus, remember, existing here before it copied, quickly entered M phase, even though it wasn't ready to do so. And finally, we should write it here just to be complete. When you take this M phase cell and you fuse it with an S phase cell, the nucleus in the S phase at this point should not come as a surprise to you, also quickly entered M phase. And so their hypothesis was there must be a cytoplasmic molecule that controls M phase entry. That some molecule in this M phase cytoplasm, when it is transferred to the G1 cytoplasm, causes it G2 cytoplasm or G1 cytoplasm or S cytoplasm, was enough to send it into M phase, that there was some kind of protein regulating this. So now to test this hypothesis, they wanted to take cytoplasm from an M phase cell and transfer it directly into a G2 phase cell to make sure it really was some kind of protein in the cytoplasm. So what they did was they took a cell that was stuck in M phase, and it contained the nucleus, of course, but what they did was they removed the cytoplasm from here. So let's just make a tube here full of cytoplasm from this M phase cell. They then took a cell that was in G2. Remember last time they just fused them together. But now what they're going to do is they're going to take this cytoplasm that they just isolated from the M phase cell and place it into this G2 cell. They also took cytoplasm from a cell in interphase. It could have been G2, but just any interphase cell. They removed that cytoplasm, and they also then put that into a G2 cell. If the experiment was going to support their hypothesis, what they would see is this G2 cell move into M phase. Now also, to support their hypothesis, what they would see when they transferred interphase cytoplasm to the G2 cell is you would see the cell remain in G2. So if their hypothesis is supported, what they would see is the cell G2 cell transfer into M phase by the addition of M phase cytoplasm, meaning there was some kind of protein found in this cytoplasm that was enough to trigger this. Likewise, by just adding more cytoplasm from G2, that should not be enough to send it to M phase. If they saw these two results, that would support their hypothesis. And that is what they saw. They saw no transition to M phase in the bottom, and they saw transition to M phase on the top. After this was shown, a lot of research was done to identify the protein that was necessary to send the cell from G2 into M phase. And so they analyzed this cytoplasm here, and they discovered a protein factor called M phase promoting factor. And we're just going to call this from here on MPF. And now I'm going to move on to the next whiteboard, and we're going to talk more about MPF, what it contains, how it becomes active, how it becomes inactive. Okay, on the last whiteboard, I explained to you how researchers were able to identify MPF and how this MPF protein would cause a cell at G2 to move into M phase and stimulate all the events associated with M phase. The condensing of the chromosomes, the lining them up during metaphase, then pulling them apart during anaphase and telophase. So once they identified this protein, they wanted to learn a little bit more about it. And one of the first things they were able to characterize is that it consisted of two subunits. This one over here, we're going to call CDK, and this stands for cyclin dependent kinase. And over here, this other subunit is called cyclin. It is the cyclin that's associated with the cyclin-dependent kinase. Since it has this kinase, and we know that kinases add phosphate groups to targets, and when they add phosphate groups to targets, they are typically associated with activation, but they are also associated with inhibition as well. Most of the examples we've talked about have been linked to activation, but sometimes the phosphate group can be added and it inhibits the enzyme. They reasoned that this kinase probably was the thing that was important during M phase, that it likely added phosphates to activate or inhibit certain processes necessary for M phase. But what they discovered was that the concentration of CDK was constant throughout the cell cycle. So if we were to draw this table here, and we'll put the phases of the cell cycle here. Over here, let's put enzyme concentration. They predicted that CDK would be the highest during M phase, because that's when the kinase activity is going to be important to target certain events in the M phase. So they reasoned that that would increase in concentration at M phase. But as is true with most good ideas, they were wrong. And so during this time of M phase, which we can kind of highlight here, the concentration of CDK, so the blue line is CDK, was the same as it was in G1S or G2. In fact, if you look at any part of the cell cycle, it's going to have about the same amount of CDK. But they knew that at M phase, the CDK became active. G1S and G2, it was inactive. So let's make a little key over here. This means active. CDK was active here, yet it was present throughout the whole cell cycle. So how did it become active here but not throughout G1S and G2? And that's when we're going to come back to our other subunit of the MPF. The concentration of cyclin steadily increases between G1S and G2 until it reaches this critical concentration at M phase. And then it will slowly decrease during M phase. And then cyclin will once again increase again to the next M phase. And then it will decrease throughout mitosis. We should point out a few things here. Because remember, we don't want any activity. We don't want mitosis to begin prematurely during interphase between G1S and G2. And so the cyclins that bind to CDK early on don't activate it. And so let's talk about how we can keep this inactive until the beginning of M phase. So let's talk about how it's going to remain inactive during interphase. So let's draw the enzyme, the MPF here, with the CDK and the cyclin protein subunits. During this state, even when they are bound like this, because as the concentration increases, they will begin to bind before M phase. But before you hit M phase, it's phosphorylated at two sites on CDK here. And as long as both sites are phosphorylated, it remains inactive. So this is inactive MPF. As it approaches M phase, what happens is this phosphate group is removed. So at this stage here, during mitosis, that phosphate group is removed. And you only have this one phosphate group on it. One phosphate group on it, it is active. And it only needs to be active during the first part of mitosis because that sets in path a series of events that will trigger the condensation of the chromosomes, the forming of the spindles, and so forth. Once it's initiated that pathway, the activity of MPF slowly decreases. And it decreases, as you might guess, because you can see cyclin decreasing in concentration. So over time, what's happening is the cyclin portion of MPF gets ubiquitinated. And you can just put UB here. But it adds a protein molecule called ubiquitin to the cyclin. When cyclin is ubiquitinated, it is slowly degraded. So during G1, you're going to just have the single subunit of MPF, the CDK. And then, as this next cycle begins to ramp up, cyclin once again will become expressed. And once again, it's going to bind to this inactive CDK. And then once it hits M phase, it'll lose that phosphate group. So we can draw this real quick here. So during S and G2, you can see that cyclin once again will start to accumulate, and once again, they will bind to each other. But they'll remain inactive because of these two phosphate groups. Once they hit mitosis, M phase, that phosphate group is removed, and it is once again active. And the whole cycle begins again. Cyclin begins to degrade, and then cyclin begins to increase in the next cell cycle. So it's this combination of this constant supply of CDK being regulated by the increased expression of cyclin, then the decreased expression of cyclin. Increase, decrease. So what I have described for you is one protein factor that helps regulate one part of the cell cycle right here. And what we've learned over the years is that there are other places along this cell cycle that are also regulated. In fact, many of them are regulated by other cyclin-dependent kinases and other cyclins. So now I want to briefly talk about the three main cell cycle checkpoints. Not at this level of detail, but just in a general sense. Okay, we have the G1 checkpoint. We have the G2 checkpoint. And we have the M checkpoint, which really has two components to it. The G1 checkpoint exists right here. And so the cell, when it's in G1, it cannot go to S phase until it's past this checkpoint. And so what happens at this checkpoint that this cell has to pass? Well, it needs to check to make sure in general that the cell is ready to enter into a new round of division, to copy its DNA and then to form the two new cells. And so it checks its size to make sure the cell is the right size to proceed. Because remember, as we move through the cell cycle, we're going to produce two new cells that are going to be about the same size as this cell. So as we move forward, this cell has to get bigger. Nutrient availability. If there aren't proper nutrients available, the cell will not move forward. No reason to make two new cells if nutrients are poor. Unicellular organisms like yeast and bacteria use this quite efficiently. If the nutrients are poor, they will stop right in G1 and they won't move any further. 3. This is social signals. We think of this more in multicellular organisms. But thinking about quorum sensing, you could make an argument for social signaling in unicellular organisms as well. But essentially what this is saying, in a multicellular organism, is this cell receiving signals from other cells? Is it receiving signals, say, from the pituitary gland, from the adrenal gland, or some other gland in the body? Is it receiving that signal that says it's time for it to divide? So that's what we mean by social signals. Cells from other parts of the body. This fourth one involves DNA damage. Proteins in this cell will assess whether or not there's DNA damage. And if there's damage to the DNA, it will do one of two things. It will either repair the damage, or it will have the cell enter apoptosis and kill the cell. If all of these requirements are not met, the cell will stay in G1. It will not enter S phase. Now, as we talk about cancer, there are mutations that can occur that will permit the cell to ignore this checkpoint and move forward. So you could imagine if there is DNA damage present, yet it ignores that checkpoint, those pieces of the DNA that are damaged here will continue to exist as the cell is dividing. And the new cells will have that DNA damage, and then their cells will, and their cells will as you move forward. So blowing past this stop sign here can have damaging effects to the cell and ultimately to the whole organism. At the G2 checkpoint, the two main things it's going to check is the DNA fully copied? Because you don't want to enter into M phase unless it's all copied. I should mention, it might have been implied, but I should mention that the G2 checkpoint exists right before you enter M phase. Okay, so it checks to see if the DNA is fully copied, and then it checks for mutations. It wasn't surprising for them to discover that the MPF proteins that we just talked about, the cyclin-dependent kinases with its related cyclin, are key regulators of this checkpoint. In fact, one key way to do this is if the DNA is not fully copied or if there are mutations present, MPF, that's CDK portion of it, will remain phosphorylated at both sites here, the activating site and the inhibitory site. So as I mentioned before, there are two parts to this here. The first one is the spindle attachment. Remember, you have your cell here in mitosis, and you have these centrosomes here that send out these spindles that attach to chromosomes. Before a cell can move past this checkpoint, move past metaphase here, all of these spindles have to be attached to a chromosome. If they're not attached, the cell cycle will stop right here. If there's an error here and it goes past this checkpoint without them being attached, you can end up with cells that have fewer or more chromosomes in them. The other part of the M cycle checkpoint involves cyclin degradation. That transition from anaphase to telophase requires the cyclin protein to be degraded. And as long as cyclin is present, it will not move forward in the cell cycle. Okay, that's all I want to say for this podcast. In this podcast, we talked about why it was important that this cell cycle is regulated. We talked a lot about how MPF was involved in this transition from G2 to M and the experiments that led to that discovery. And then we closed with the discussion of these 3 checkpoints. So we closed with these 3 checkpoints, and we talked about why it was important that these cell cycles checkpoints exist to stop the cell until it was ready to proceed. We hinted a little bit on what would happen if these cell cycle checkpoints were not followed. And in the next podcast, that's what we're going to talk about. We're going to talk about how when these cell cycle checkpoints fail, how that can lead to cancer. All right, if you have any questions about the material in this first podcast, let me know. If not, I'll see you in class. Bye.